real-time PCR is really fantastic. The instruments that can do real-time PCR, what they're doing is they're monitoring how much product is in the tube constantly throughout the run. And so with the kind of PCR that we've been doing in class, that's called endpoint PCR, or just conventional PCR. So conventional endpoint PCR. We run the PCR for 30 or 40 cycles, and then we run it on a gel, and what we're seeing is the end product after the 30 or 40 cycles. So PCR is great, it's super sensitive, and it gives us a yes or no answer on whether we got our product or not. And we already did this with our first RT-PCR experiment on the TTR gene for mouse, where we just asked the question, is the TTR gene expressed in adult mouse liver? And our conclusion from your RT-PCR experiment, which worked for all of you, is that yes, the TTR gene is expressed in mouse liver, because we all got that band at 150 base pairs. You all remember that? Mm-hmm. Okay, great. Doing good, even after a night of debauchery last night. Anyway, so conventional PCR for gene expression can give us a yes or no answer, but that's it. Real-time PCR can give us this real-time information that we can use to calculate how many starting copies we had of a particular DNA or RNA. So for gene expression, the great thing about this is we can use real-time to say, this is how much we started with, right? Instead of just looking at how much we have at the end, it can tell us how much we had to start with. So I have to take you through how this works. So, real-time quantitative PCR gives us curves that look like this, and we'll talk about this for the next little bit here. All right. So, how are we going to quantify our PCR product? We want to use PCR to somehow figure out how much material we started with. So in gene expression, we're going to run PCR, and then we got to figure out how that can tell us what was in the cells originally. So to do that, we have to understand the math of PCR a little bit. It's really very simple. So here, P is the amount of PCR products that you have. It's doubling every cycle. The number of cycles is n. So 2 to the n would be the amount of product you'd expect after n number of cycles if you started with one molecule, right? So P would equal 2 to the n if we started with one template molecule. Well, we don't ever start, or rarely start, with just one template molecule. We're starting typically with hundreds or thousands. So really the formula needs to be the amount of product equals T, which is the starting quantity of the template, times 2 to the n. So doubling every cycle, but we're starting with more than one copy, right? Make sense to everybody? Seems like after last night doing math with you first thing in the morning is cruel. So that's what we're but one of the things about PCR is that as you go along in the cycles, you're not getting 100% efficiency at every cycle. So, at the first cycle, you might get 100% efficiency, but then at the next cycle, 98%. And then 97%, and then 96%, and then 95%. So we also have to include efficiency. So if we really wanted to understand how much product we'd expect at the end, we'd have to have the amount of starting material, and then instead of this number being 2, because it's doubling every cycle, it would be something less than 2. So if the efficiency was 90%, for example, this would be 1.9. So it would be 1.9 to the n if the efficiency was 90%. Does that make sense to everybody? The problem is if the efficiency is changing every cycle, this formula doesn't work, but it just gives you an idea of what we're up against, right? Now, the efficiency makes a huge difference because, okay, so here we go. Same thing I just said. In the next slide, I'm going to show you the effect of 10, 20, and 30% decrease in efficiency for the PCR. So if you have 100% efficiency and you start with one copy, by the time you get to cycle 30, you've got 1 billion molecules, okay? If you have 90% efficiency by the time you get to cycle 30, you've only got 230 million. If you have 80% efficiency, 45 million, 70% efficiency, 8 million. So the efficiency is going to have a dramatic effect on the amount of product, and so efficiency is a huge problem for us in trying to be quantitative. And the reason is, what we're going to try to do is we're going to try to use the amount of product that we get from the PCR to back calculate how much we started with, right? Back calculate how much we started with. So because of this efficiency issue, that's problematic, right? It turns out that with PCR, for about the first 10 cycles or so, and maybe after 15 cycles, you're really close to 100% efficiency. You're close to 100% efficiency. Because of that, if we monitor how much product we're getting at each cycle during those early cycles, we actually can back calculate how much template we started with. Okay, we can back calculate how much template we started with. So let me go back to here. If we are using the early cycles, we can forget the efficiency, and we can just use this formula, where we solve for T, which was our starting quantity of template, right? So if we solve for T here, and we know the amount of product we have, then we can get our starting quantity. So the instrument does this for you. You don't actually have to do the math yourself. The trick is that you have to be looking at your amount of product in those very early cycles where you're close to 100%. If you know you're doubling every cycle, and you measure the amount of product at cycle 10, you can back calculate how much you have at cycle 0, right? So that's what we're doing for real-time PCR. If we have 1,000 copies at cycle 10, then we know that at cycle 9 we had 500 copies, right? And we know that at cycle 8 we had 250 copies, and we know at cycle 7 we had 125 copies. So you can back calculate because you know you're doubling every cycle. We can't measure the amount of template at the beginning because it's too small, but after we get up to 10, 12 cycles, the instrument can read how much product there is, and then we can back calculate, okay? So it's pretty cool, but it depends on the instrument being able to read these very low quantities of product when the PCR is still being 100% efficient, okay? When the PCR is being 100% efficient, we're doubling every cycle, right? We know that, then we can go backwards to figure out how much we started with, right? Make sense? It's pretty cool, and it works. It's amazing. Okay. Yeah, it's going to be a little bit different depending on how pure your DNA is and so on, and that's why you don't always see the exact same shape on your curve. So we'll take a look at that. Yeah.
How do you actually physically count the number of product molecules?
Coming. Okay. All right. So the basics of qPCR: the real-time PCR process collects data at time points throughout the PCR process. So we're not just looking at the end. We're looking at the amount of product constantly during the run. It's done by measuring the fluorescence in the wells of a PCR microtiter plate. The way this works is that there's a reagent that's in the reaction that gives us fluorescence only when we have product. amount of fluorescence is going to be proportional to the amount of product. So we're gonna use fluorescence as our indicator of how much product we have, how many molecules we have. The amount of fluorescence during the exponential phase of the PCR correlates with the starting copy number, and that's how we can do that calculation. And so we can get quantitative data from this. The higher the starting copy number, the sooner the fluorescence will be observed above background, right? If you start with 1,000 copies, you're going to get to a point where the instrument can read your amount of product faster than if you start with 10 copies, right? Because remember, we're doubling every cycle. If you start with more, you're going to get to that threshold faster than if you start with less. There's a threshold at which the instrument can start reading how much product you have, and that's not at cycle 1 or 2 or 3, it's up around cycle 10, 11, 12, 13, 14. But that's still better than we can do by eye, like you can tell. Okay? Any questions about this?
So what is the definition of a copy number?
Oh, the definition of copy number?
Copy number of a gene?
So what we're really thinking about is, for example, for our experiment where we're looking at RNA, we're thinking about how many copies of that messenger RNA did we start with that then got converted to cDNA, and then we started doubling. So we're back calculating how many original cDNA molecules we had, and therefore the number of RNA molecules that we have. Okay, does that make sense?
And the fluorescence is just so far that it has this region.
Yeah, I'm going to take you through that in more detail because we're going to use two different methods. Again, we're not going to take the easy way out here. We're going to show you two ways of doing it. Okay? The two most common ways of doing this, the class is going to do both. We're actually going to have one lab do one and one lab do the other. Yeah. Yes. So as soon as we get all your email addresses today to send you your 8-colony PCR gel, we're also going to send you the lectures. But we need all your email addresses before that. Okay. All right. These are images I got from Applied Biosystems, so they're not mine. I apologize for the fact that they're a little bit crude. But this is what your curve's going to look like. I'll show you better curves later. But what's going to happen when you do PCR? So the number of cycles is the x-axis here. The y-axis is the log of your number of DNA products. And that's, you're not actually looking at the number of DNA products, you're looking at fluorescence here. Okay? As our method for measuring how much DNA we have. Now in the early cycles, again, you're going to get doubling every cycle, so that's this green phase, and this is called the exponential phase, and this is where things are really precise, and as long as we're in this exponential phase, if we measure how much product we have here, we can back calculate how much we started with here, okay? As long as we're in that exponential phase, because we know that at every cycle we're doubling. So if it was 1,000 here, it's 500, it's 250, it's 125, etc. Why are we not stopping here? Well, because before we start the run, we don't really know where that exponential phase is going to start and stop. It's going to be different reactions. So we let the thing run, and it's actually interesting for you to see those results. We could stop them sooner than we do, but what's going to happen is eventually you go out of that exponential phase, and then the curve starts to round off, and then eventually you get a plateau where you're just not making any more product. So here we're getting less efficiency, less and less and less and less efficient, and then finally you hit a plateau where you're not making any more product. Now, why does a PCR reaction become less efficient eventually, and why does it finally plateau? It's not because you're running out of reagents. It's not because the enzyme is, pooping out after 20 cycles or so. It's because you're generating so much product that as you start doing the PCR, say it's cycle 15 or whatever, you're denaturing your product to then hybridize primers. But now you've got so much product that the products want to hybridize together. And so your products hybridizing back together start out competing with primers. And the product out competing with primers is why you start getting less and less efficient, right? And the farther you go, the worse it gets because you're getting more and more product. And finally, when you get out to the plateau phase, the product is winning all the time. The primers just don't get a chance to get in there to make more copies. Does that make sense to everybody? Good. Okay. Move on. Here's an example of an actual experiment from my lab. We're looking at two different viruses in a blood sample. So we're looking at RNA viruses. So we're doing first making cDNA from the RNA and then doing the PCR. There are two different viruses here in the sample. This is the number of cycles across the bottom. This is the fluorescence. So the question is, which of these two viruses, A or B, is present in higher concentration in the sample? Is it A or is it B?
Okay.
It's A because A is coming up faster, right? Because there are more starting copies of A, it's amplifying more quickly and crossing the threshold sooner than this other virus here, which is a lower copy number. This one's taking a lot more cycles before we finally cross the threshold and get our reading. So the threshold is set by the instrument. The instrument sets a threshold where it's reading the signal, but it also knows that your samples are in the exponential phase. So it's not going to read down here because it needs both samples in the exponential phase. So it's put it up here. Now you could adjust this a little bit in this range, and it would still give you the same result. But you can do it manually, but the instrument does it for you, so we don't have to worry about where to set that threshold. We can even look at this and say something about what the relative quantity is of these two viruses in the sample. So A, these are replicates. Okay, these are replicates. If you look at A is crossing the threshold at about cycle 22, right? Cycle 22. And B, let's call that 32. We'll average these samples of 32. So that's about a 10 cycle difference. 10 cycle difference is a lot because at every cycle you're doubling. So that's going to be 2 to the 10th difference in the amount of starting material of these two samples, 2 to the 10th. So times 2 is 4, times 2 is 8, 16, 32, 64, 128, 256, 512, 1024. So about 1,000-fold difference. So what we would conclude from this experiment, without doing any fancy math, is that there's about 1,000 times more of this virus than that virus. And that's the kind of work that we do with quantitative PCR. We're always doing, not always, but almost always doing a relative comparison. We're saying there's 1,000 times more of this than there is of that, or there's 1,000 times more of this than there is of this control that we put in. We put in a control and we know how much control we put in. And this one is 1,000 times more than we know how much of this there was, right? Everybody get that? This is a control instead of another unknown sample. Then we can figure out how much we had of this unit. Okay? So pretty cool. So we can do relative comparisons like this, or we can get actual estimates of quantity. Okay, so that's just an example, and it shows you the basics of the calculation. Now the instrument actually measures where things are crossing the threshold. So I said 22 here, but the instrument might do it, come up with 22.2, and it'll do the same thing over here, and then it does the math for you and spits out. class, but the instrument's gonna do it for us. Okay. So, types of quantitative PCR. You can do absolute quantitation if you include a standard, and you can do a standard curve, or you can include a couple of standards in your run to allow you to calculate absolute quantitation. And Nils will talk about this more next week, so don't worry too much about the details. Or you can just do relative quantitation, i.e. the amount of virus in one sample compared to another sample or to a control. Relative quantitation is mostly what you see in the literature. It's mostly what's published. In our experiment, for example, we're going to look at the expression of the PCR gene in adult mouse liver, adult mouse spleen, and adult heart. And what we're going to do is look at the relative expression in those three tissues. If we ran the appropriate controls, we could also do absolute quantitation, but mostly people don't do that. Mostly what's published is relative quantitation. But you can do both. You can do both. Okay? Seem okay? All right, again, we're gonna go through this again next week, so if any of this is a little fuzzy, don't worry. We're gonna do it again when you get your results. All right, what are some of the advantages of real-time PCR detection? Again, real-time because it's monitoring your amount of product constantly throughout the run. So the automatic detection of PCR products throughout the amplification process is really useful. We're going to collect our exponential fold data. By doing this, we're going to know where in the run everything is exponential, so that we can do that back calculating. This kind of RT-PCR can reliably detect twofold or less difference in starting template. 1.3 or 1.2-fold difference can also be reliable if you do replicate. And so people publish this kind of data all the time. People will publish and claim significant differences with less than a twofold difference, but you've got to do a number of replicas. One huge advantage to this is you're not doing any post-PCR steps. You don't have to take your product out of the wells and run a gel. These plates are sealed, and so once your run is over and you have your results, you can just get rid of your plate without ever having to open it up, and that reduces lab contamination. So that's a big advantage to real-time PCR over standard conventional PCR, where we have to load our samples on a gel. Okay? So you're much less likely to contaminate your lab when you're doing this kind of work compared to conventional PCR. Okay. All right, there are two real-time PCR chemistries that are in use, and we're going to do both of them. One is an approach using cyber, which you've already heard about because we stain our gels in cyber green, so this is the same dye. It works by binding to double-stranded DNA, and it's called an intercalating dye because what it does is double-stranded DNA is it inserts itself into the helix. It intercalates between the two strands. And so cyber is referred to as an intercalated dye. And so that's one way we're going to do it. We're going to use a dye that only binds to double-stranded DNA, not single-stranded. And so we're going to be able to see our increase in product by the amount of fluorescence from the cyber. Okay, so that's one method. The other method uses a probe, and this method is significantly more expensive because these probes cost money, but it's a great method that we use all the time. We use this in our diagnostics, and this is called the TACMAN assay, or here at Smith we would refer to this as the TAC person assay, or maybe the TAC woman assay. So there we go. All right, what are these two approaches and how they work? How do they work? Well, here's the cyber green approach. So imagine we're doing PCR, we've denatured our two strands of template, And now we're doing a PCR amplification with a primer. So this represents our primer, it's annealed to our template. This is our polymerase here, and it's tracking along the DNA and copying. The little red blobs here represent cyber. They should be green blobs because cyber green, right? But they're red in this picture. And when they're in solution like this, they don't fluoresce. But when they bind to the DNA, when they interpolate, then they do fluoresce. And so that's what the little burst here indicates, is that now we're getting fluorescence. So the more you copy, the more fluorescence you get. The more copies you make, the more fluorescence you get. Okay? So this is the way that the cyber green works, and it's definitely less expensive than the TACMAN approach, but the specificity is lower here because anything that amplifies is going to give you a signal. So if this is some kind of off-target amplification, it's still going to give you a signal. You're going to see with the TACMAN approach that we have better specificity. Now, if your primers are really good and you're only amplifying the product you want, then the cyber green approach should be just as good. And we're going to show you that in this experiment. Okay, the TACMAN system is also called a 5' nuclease assay, or the TACMAN assay. It was developed by Applied Biosystems. It uses a probe that we call a TACMAN probe that allows us to detect the amplification in real time. And the TACMAN assay uses the 5' nuclease activity of Taq polymerase. So Taq polymerase synthesizes 5' to 3', but it also has a 5' to 3' nuclease activity. And I'm going to show you that in the next slide. And this 5' nuclease activity is important to the way that the TACMAN system works.
You have to do that because you then have to know what you're testing when you test the probe.
Yeah, you need to know the sequence of the gene that you're doing the real-time PCR on. For both systems, you need to know the sequence of your target because you have to design primers that are specific to the target. With the TACMAN system, not only are you designing a forward and reverse primer, but you're also designing the probe. You're designing 3 oligonucleotides for the TACMAN system. Well, typically when you are doing gene expression studies, so for example in our experiment we're doing PCR, the whole mouse genome sequence is known, the sequence of the TTR cDNA is known. You need to have that information before you can design an experiment like this. Okay, so for a lot of the common organisms, this isn't an issue. If you work on weird infectious agents like we do in my lab, then you've got to sequence your own genome so you know what you're dealing with. So it's easy for most researchers. If you work on weird organisms, then it becomes more difficult. Okay? So this shows how the Taq polymerase works. So this is really a weird diagram from Applied Biosystems again, so let me explain what we're looking at. This represents one strand of the double helix. This would be the primer that's hybridized. So this would be the 5' end of the primer down here. This would be the 3' end. This is the Taq polymerase here. And what the Taq polymerase is doing is it's synthesizing 5' to 3' this way, so it's moving in this direction. This part of the polymerase is the part that's doing the synthesis, that is adding the nucleotides to the 3' end here. Here's the next nucleotide coming in, right? It will be added there. Over on this end, this is our probe, and the probe is going to get degraded by this 5' nuclease. The 5' nuclease is the front of the enzyme. The synthesis part of the enzyme is the backside of the enzyme here. So while this is adding DNA to the 3' end, this part of the enzyme can be degrading DNA in front of it. And so this is a 5' exonuclease, exo meaning that it starts from an end and takes off one nucleotide at a time. And this is referred to as the 5' to 3' exonuclease activity. Restriction enzymes are endonucleases. They cut within a molecule. So those are endonucleases. This is an exonuclease because it's starting from an end and it's cleaving on one nucleotide at a time. Okay? So, Taq polymerase has both 5' to 3' synthesis activity and a 5' to 3' exo activity. Okay? So, the staff always likes me to do my little dance for this. So, the idea is that if I'm Taq polymerase, I have my 5' to 3' synthesis activity back here, so I can add nucleotides back here. Meanwhile, I can be cutting off nucleotides going in this direction, right? So, why do cells have enzymes like this? Well, you need to have enzymes like this in order to repair DNA where mistakes have been made, for example. So, polymerases that have both 5' to 3' synthesis activity and 5' to 3' exo activity are pretty common. So, this is the enzyme that we're going to be using in this assay, and we're going to be using this 5' to 3' nuclease activity. So that's why I needed to tell you about this before rolling forward here. Any questions about this?
So the primer is right here.
This is the primer,
And
and this is the probe.
the probe.
Yeah,
is
this will be
the-
more, I have better diagrams in a second. So,
Okay.
yeah, so let me just show you the next one, we'll see if it clarifies. So, here are our two primers for the PCR, and this would be your forward primer, this would be the reverse. These are our two template strands, right? And now we have a third molecule in here, a third short molecule, and that's the probe. The probe is about the same length as the primer, so we're in the range of 20 to 25 nucleotides in length. The probe has on it a reporter dye at one end. This is going to be a fluorescent dye, and it's got a quencher molecule at the other end. Now, when the two are together, they're floating around in solution. The quencher is keeping the reporter from putting out a signal. And the physical chemistry here is known as FRET, fluorescent resonance emission. Oh, electron transfer?
Energy,
Okay.
energy.
Energy transfer.
Energy transfer, fluorescent resonance
It's actually
energy.
Forster, but.
Anyway, I had physical chemistry about 1,000 years ago, so, anyway. So it's inhibiting, the quencher is inhibiting the reporter dye from fluorescing. And the same thing is true when it's hybridized to the template. These things are too close together, and so the reporter does not emit the signal here. Now, imagine that the Taq polymerase gets on here and here, and it starts copying. It's going to copy along here, and then it's going to get to the probe, and it's going to start lopping off nucleotides, right? And when it takes off this first one, it releases the reporter, and now the reporter is going to emit a signal. Okay? It's going to emit a signal because it gets away from the quencher. In our experiments, we use probes that have two quenchers. There's a quencher on the end, and we put a quencher in the middle to really keep this thing shut down. So we actually use two quenchers, but that's not important for right now. Okay, so here's our probe on here. There's our reporter. There's our quencher. So here's the forward primer. FRET is keeping the reporter quiet here. And then we're going to do polymerization off of our two primers. So this one goes this way, and that one goes that way. And as it degrades the probe with that 5' to 3' exo, the reporter is released and we get a signal. And the amount of signal that's released is proportional to the amount of product that we're producing, because we give one signal for every molecule that gets copied. Right? Every DNA double helix that gets copied gives us one emission.
So even in the top diagram, the report is not anything.
Not emitting yet, not emitting until the cleavage occurs and then it's emitting.
So the probe has to be the very end of
The probe is always on the recorder is always on the 5' end of the probe, and the quencher is on the 3' end. We also put a quencher in the middle.
but how do we know that it's a complete amplifon?
How do we know that we go all the way through to the end?
Yes.
The PCR that we're doing here, we always keep the primers pretty close together. Very common would be somewhere between 100 and 150 base pairs. So these are really close. So that enhances the probability that our molecules are all going to the end. Yeah. This 5' to 3' exo activity is really efficient. Really efficient.
So the reverse primer,
There's-
does not from the probe.
No, the probe only hybridizes on one of the two strands. So for every double helix that you copy, you make one. Okay? For every double helix we copy, we make one. Okay? I suppose you could design a second probe that would hybridize to the other strand, but nobody does that. Okay?
Is there an efficiency associated with the activation of the reporter? So if it's not 100%, then that would affect the calculation as well, right?
So my guess is, I'm not a physical chemist, and I don't know what the concentrations would have to be, but my guess is, if you do too many cycles, or you get way out there, you're gonna start releasing so much quencher that then there's gonna be quencher out there inhibiting the signal. And so my guess is that, but that's gonna be past the point where we're beyond the exponential amplification anyway. So it's not a concern for going forward. Okay? So, this is just another cartoon, just so you've got multiple images. Again, now we're just showing one strand. So this is the 3' end at this end and 5' end at this end. This would be the probe hybridized. And then here's our primer hybridizing with the polymerase, and we go along and we degrade this, and then the reporter can give a signal. So it's just showing the exact same thing I did in the previous one, just another cartoon to give you another visual. Any questions about this? So the method by which we're getting a signal here is fluorescence, but it's involving this third molecule, this probe. Let me go back to this one. So we're getting specificity of this PCR from both primers, but now we're also getting specificity from the probe. Three things have to hybridize here in order to get a signal, right? So that's why the TACMAN system is considered to have better specificity, because not only do you have to have your primers matched, this thing has to match. And so that decreases the possibility of getting any off-target signal. And so if you ask people which of these two systems is better, the cyber system or the TACMAN system, most people will say, well, the TACMAN has this inherent advantage of having this third molecule giving you specificity. But these probes that are designed with the reporter at one end and the quencher here, and now we always use one with a quencher in the middle, these are not cheap. And so this kind of assay is more expensive. So it's got an advantage, it also has the drawback of cost. And so, you know, which method you use might depend on your budget. Now, what I'm here to tell you is that if your primers are really specific and you only get one product, then the two methods give you the same result. And we're going to have one of the labs do cyber today and one of the labs do the TACMAN system. The PCRs are set up exactly the same. So there's no difference really in your protocols, except for a very minor change, and that's that this one includes a probe and the other one doesn't. And in the end, we're gonna show you that because our primers are so good that it doesn't matter which one you use. So if you're in the cyber green lab, don't feel like you're getting shortchanged. Your results are gonna be just as good as the TACMAIL one. The reason we're doing both is we wanna show you that. We wanna show you that they both work, okay? All right. Okay, this is just a little cartoon to remind me to tell you that the way the instrument works is that, and you're gonna see the instrument later, it's called a quantitative PCR instrument. It's bigger than a regular thermocycler, it's more complicated, because what this instrument does is it shines light of a specific wavelength onto your sample. Now this is a tube, but what we're gonna do is load all your samples into a 96 well plate, and in the 96 well plate, your samples will be arrayed, and we'll know which sample is in which well, and these instruments shine this specific wavelength of light onto each specific well of the 96 well plate. So these 96 well plates have to be optically clear. These are special plates, and when the light is excites your samples, your floor emits light of a specific wavelength that is then picked up by a detector. So these instruments are pretty fancy because they have to focus light on each of the 96 wells and then has to read each of the 96 wells. So pretty nifty instruments. The cost on these used to be quite high, and now the cost keeps coming down, which is nice. And so we'll show you these instruments. The other thing is the instruments used to be really big. It's getting smaller, which is also nice. Okay. Yeah. So I'm gonna stop there. That's my introduction. When you get your results next week, and you've got your results in front of you, and we're showing the results from both